Hi, so I'm going to talk about a phenomenon called genetic nurture, and this is when the genotypes of family members influence the trait of a phenotyped individual through the environment. And this is something that Augie and I have been investigating, primarily with the decode data, and we think it's a very important phenomenon for understanding a lot of the sort of social and behavioral traits that people here are interested in. So the primary form of genetic nurture that we've been investigating is parental genetic nurture. So this is when the genotypes of the parents are influencing their offspring through the environment. And genetic nurture is a special case of a more general kind of effect called an indirect genetic effect. So these have been studied in animal breeding, but I think in human genetics, their influence and the consequences they have for both association studies and heritability have been underappreciated. So it's important to distinguish direct genetic effects, which are typically what we think of when we study the genetic effect of a trait. So if you inherit an allele, it has some biochemical effect on you, and that thereby affects your trait. That's a direct genetic effect. But any allele that you inherited was also present in your parents. So if it affected your trait, it could have affected your parents' traits. And by affecting your parents' traits, it can affect the environment through which you develop and affect your trait in this more indirect path. And what that means is that if you're just looking at correlations between the alleles of individuals and their traits, that correlation can be driven not only by the direct effect, but also by the indirect effect through the family. And this is something that we investigated for educational attainment in Iceland, as I'm going to talk about in more detail. However, you can have this kind of genetic nurture or indirect effects from other relatives. So siblings could influence each other. I think one area where siblings might have a particularly strong effect is in some of these addictive behaviors, such as smoking and alcohol. Grandparents can have an effect on the parents, and then the parents affect the offspring, but they could also directly affect the offspring, especially if someone was raised by the grandparents, for example. And you can have indirect genetic effects from any individuals that can be your friends, but it's the indirect effects from relatives that are most important for the study of these kind of traits, because one's relatives' genotypes are correlated with your own. So whenever you're doing an analysis that looks at correlations between genotype and phenotype, it's worth remembering that genotype is correlated with the relatives' genotypes. So there can be contributions from these indirect effects from relatives that are underappreciated without some sort of family data that allows you to separate them. And to separate them, it's easiest when you have genetic information on the parents. So there's a classical test called the transmission disequilibrium test that does this, although it was devised originally for finding genes associated with disease when people were worried about population stratification. And it deals with both population stratification and indirect genetic effects from relatives. And the idea here is that if you condition on the genotype of the parents, then what remains in the variation in the genotype of the offspring is due to the random Mendelian segregations in the parents. And those random segregations in the parents are independent of environmental effects. So you can estimate the direct effect of inheriting the allele in a way that is robust to environmental confounding, both indirect genetic effects and population stratification. So the TDT works by saying if you have parents that are heterozygous at a SNP, say AT, then by the laws of Mendelian inheritance, you expect 50% of the offspring to have the A, 50% to have the T. But if, say, the A actually, it contributes to causing this disease, the A allele, then within offspring that have that disease, you would expect overtransmission of that A allele. And that kind of a test is incredibly robust and was used very successfully to find disease genes in the sort of pre-GWAS era. For quantitative traits, it's a little bit different, but the sort of approach that we took in Iceland is very similar to the TDT in that you regress a trait on both the proband, the phenotyped individual's genotype, and the genotype of the parents. And by jointly regressing on the proband and the parental genotypes, you use the information in the proband genotype that comes from the random segregations in the parents and is independent of environmental effects, including indirect genetic effects. And that allows you to separate out the direct effect component from the indirect component coming from the family. So this is what we did in this paper published earlier this year in Science, and Augie Kong, who's here, really led this work. And I'm just going to go through some of the main results of this paper. So we took advantage of the GWAS of educational attainment that was published in 2016. So this isn't the most recent one. So the R squared is somewhat lower than the current state of the art. And by constructing a score, excluding Icelandic data, we sort of replicated the main result that if you simply correlate the polygenic score with educational attainment in Iceland, it explains the R squared is 4.98% here. But by doing this thing where you're fitting both the parental and the proband genotypes jointly to estimate the direct component, you see that the variance explained by the direct component alone is only around 2.45%. So a substantial fraction of the predictive power of the educational attainment polygenic score is actually coming from the indirect effects from the relatives, and also perhaps some contribution from assortative mating. And then using a procedure that, so in this third column on the right, we used a procedure that accounted for assortative mating to try and estimate the effect, the indirect effect from the genetic nurturing effect from the parents, and compare the size of that to the direct effect. So for educational attainment, we see that the nurturing effect is around 1/3 the size of the direct effect, so it's fairly substantial. And because variance explained is proportional to the square, this actually amounts to a larger drop in the variance explained due to the direct effects than you might expect. And using the educational attainment score, we also look to see if the educational attainment score of the parents then predicts other traits in the offspring. And this gets at some of the issues about correlations between traits. So correlations between traits, so-called genetic correlations between traits, can actually be driven by the environment captured by the parental genotypes. So we saw evidence for this for age at first child, in that the educational attainment polygenic score from this GWAS explains just over 1% if you just age at first child, if you just do the correlation. But the actual component of that is due to the direct effect is less than 0.5%. And you actually find an even bigger ratio between the nurturing effect and the direct effect. And we see similar results for a composite health trait, that this education score, part of what is driving the predictive power of that education score on health is actually coming from the family environment. So there's no reason why genetic nurture should be symmetrical between parents. In fact, when these kinds of effects have been examined in animal breeding, they've mainly just tested for a maternal effect, because paternal investment in most animals is pretty minimal. But in humans, you might expect there to be some difference, but maybe not as drastic. And we do see evidence that there's greater contribution from the mothers than the fathers, although we did not have sufficient power to see this with statistical significance for many traits. So it may be a little higher from the mothers and from the fathers for educational attainment and age at first child. For this composite health trait, we did have statistically significant greater contribution from the mothers and the fathers to the health of the child that's coming through this educational attainment polygenic score. So we also were able to look at effects coming from the siblings. And I think this is quite an important phenomenon, because these kinds of effects can generate bias in twin estimates of heritability. So again, we did not have great precision with which to estimate the effect coming from the siblings, but we did see marginal statistical significance for this. And this is a robust way of estimating the SIB effect, because by fitting the SIB effect jointly with the parental effect, you have this same TDT type property that it's free from confounding and population stratification. And this leads to bias in the MZ twin Z design, because the MZ twin Z DZ twin design assumes that there's equal environmental similarity between monozygotic and dizygotic twins. But if part of the environmental similarity is actually driven by genetic similarity, then monozygotic twins are more genetically similar than dizygotic twins. So that necessarily biases the MZ DZ twin design. So now I'm going to talk about what this phenomenon means for defining and estimating heritability, which is the work that I've led. And I've already introduced the concept of genetic nurture and direct genetic effects, so I just want to simply say that heritability should be defined as the fraction of trait variation due to direct genetic effects alone. And this is because from the perspective of the individual whose trait is affected, the indirect effects from relatives are environmental, even though they are driven in some sense by genetics. And this is a definition of heritability that is comparable to what twin studies are trying to estimate. So I find it useful to think about heritability estimation as operating often in this space where you have phenotypic correlation on the y-axis and relatedness on the x-axis. And one of the key problems of estimating heritability is that in general, as relatedness increases, environmental similarity increases. And most methods of estimating heritability can be thought of as fitting some sort of linear relationship to subsets of relatives within this phenotypic correlation genetic relatedness space. So genetic nurture is actually the phenomenon that creates the most difficult confounding between relatedness and environmental similarity. And this is easy to see when you think about how the correlation between people's parents' alleles increases with relatedness. So as people become more related, their alleles become more similar, but necessarily their parents' alleles also become more similar. So the correlation due to genetic nurturing effects increases exactly in proportion to genetic relatedness, which is a tricky thing to separate. The only slight difference is when you go from DZ to MZ twins, you don't increase parental relatedness because it's the same parents, but if you have these kind of SIB effects, then that can generate some similar kind of increase in environmental similarity with relatedness. So just to briefly sort of review how I see some of these other methods of estimating heritability is operating in this space. The twin study estimator is comparing this similarity of MZ to DZ twins, and it does make this assumption about environmental similarity being equal between the two twin pair classes. There's always been a question of to what degree violation of those assumptions leads to overestimation. More recently, methods that have become to be called Gremel methods, such as the embodied in the software package GCTA, have become popular. And these look at some sort of high level. They can be thought of as seeing how phenotypic similarity changes with relatedness measured from SNPs for small deviations around the average relatedness. And because they exclude close relatives, this avoids bias due to certain kinds of environmental effects. But when you have genetic nurture, the similarity due to parental alleles exactly tracks relatedness, even for very distant relatives. So it picks up bias from those kinds of effects. A method that's perhaps less well known but also important is one that I call the kinship method, and this measures relatedness using identity by descent segments. And while it can control for some kinds of environmental effects by fitting additional components of the variance, it really has no way to distinguish between similarity due to genetic and environmental causes. A more robust method that was invented by Peter Vischer in 2006 looks at how phenotypic similarity changes with relatedness for sibling pairs only. And this variation in relatedness between siblings is generated by the random segregations in parents, which are independent of environmental effects, so it can estimate heritability in a very robust way, but it requires hundreds of thousands of genotype sibling pairs to get precise estimates. So when I was in Iceland, I developed a novel method for estimating heritability relating to disequilibrium regression that in some sense is a generalization of SIB regression to all relative pair classes. I think in the interest of time, I might skip some of the mathematical components, but the basic idea here is you can split the phenotypic variance not just into a genetic and environmental component, but you can split it into a genetic component, heritability, and you can split the environmental component into components that are correlated with genetics and not correlated with genetics. So it's a slightly more general decomposition of phenotypic variance, and it's typically done in quantitative genetics. And the RDR covariance model uses this variance decomposition, and then these three different relatedness matrices that come from both offspring and parents and the relatedness between those different classes of relatives. So relatedness disequilibrium regression fits this covariance model, but you don't need to worry about all the complexity of that. The key idea can be understood by understanding this figure, which explains the phenomenon of relatedness disequilibrium, which is sort of a generalization of this relatedness distribution between siblings. So not only can you measure the relatedness between a pair of individuals, you can measure the relatedness between the parents of one individual and the parents of the other individual. And that relatedness between the parents is on the x-axis, and then the relatedness between their offspring is on the y-axis. And based on the laws of Mendelian inheritance, you can calculate the expected relatedness between the offspring given the relatedness between the parents. And then the vertical deviations around this diagonal expectation line that's shown here are generated by the random segregations in those parents. So by fitting these different relatedness matrices jointly in my method, use this information in relatedness that comes from random segregations to estimate heritability, but you can use the many millions and millions of more distantly related pairs rather than just the siblings, which you get in SIB regression, which then gives you enough power to estimate heritability in a very robust way. And that's what I did and compared to some of these other methods. So this is comparing to the kinship method where you control for some element of family environment. And I just highlight the educational attainment result here because there's really a big difference between my method and this kinship method for educational attainment. And I think that's driven mainly by the phenomenon of genetic nurture. And that's important because there was this paper in molecular psychiatry this year finding a high estimate of heritability from a method very similar to the kinship method, and I think it's seriously biased by genetic nurture. So Iceland has such a small population that there aren't even enough twins to perform a proper twin study. So I compared to the Swedish twins, and I saw higher estimates for basically all the traits. I'm not sure what the explanation for this is, in that there are various possibilities, including very rare variants, overestimation by twin studies, non-additive genetic effects, assortative mating, all these sort of bogeymen of quantitative genetics that we conjure up when we can't explain something. So then I did some simulations for Gremel methods in the UK Biobank using a population of unrelated individuals. And really I'm running out of time, so I'm just going to say the key thing to take away from this is you can see that the Gremel method it's not estimating heritability as I define it and as twin studies as define it. It's estimating the total variance explained by the combined direct and indirect genetic effects from relatives, which is a meaningful parameter, but it's not necessarily the same parameter. And I can't do the standard Gremel thing in Iceland because it requires a large sample where no pair is closely related. So I have a slightly different method. It's a different inference method because if you have like 1,000 Icelanders in a room, there's going to be some close relatives, so it does pose some problems. But this method I have on the y-axis is something very similar to the kind of Gremel approach on unrelateds. And I'll just highlight here that I saw a ratio between this Gremel type method and my approach using SNPs that gives about 70% higher estimate from the Gremel type approach than my approach. And that ratio is consistent with the strength of the genetic nurturing effects we estimated using polygenic scores in Iceland. It's also consistent with within-family analyses done in the EA3 paper in different populations. So we're seeing converging lines of evidence for this phenomenon being important for educational attainment. And I'd just like to finish by acknowledging Augie especially, who's really driven a lot of this work. Thanks.
Right, good morning, everyone. I think it's the morning here, right? Thanks to the organizers for inviting me. So today I'm going to talk about a special form, or special kind of love that geneticists have, for which they found the most romantic name, assortative mating. So what is assortative mating, the name of? And well, at least when I was a kid, I liked to, the way I learned about things was by looking at differences instead of similarities. So to approach what is the concept of assortative mating, here I just shown you a list of pictures of couples, and you can see that they obviously differ in terms of height. You have couples with a short man and a tall woman, happened to be our French president a few years ago, and as well couples with different skin colors. So if that was the opposite of assortative mating, therefore assortative mating is simply the resemblance between spouses or partners. So there is long-standing evidence of partners being similar for traits like height, with a correlation ranging from 0.2 to 0.3, but also for education with the number of years is showing a correlation of or around 0.5. But those are not the only traits for which there is similarity between spouses. For example, there is evidence of correlation between partners for body mass index, for personality-related traits, strongly for political preferences, but also importantly for disease susceptibility and in particular for psychiatric disorders. So the reason why we care about assortative mating is because if we study assortative mating and its dynamics, we can understand things about different structures in the population. For example, structure that may subsequently lead to increased inequalities, for example, related to, or I could just think about if there is assortative mating on education, we can think about how that will lead to inequalities in revenues, for example. But also, quantifying assortative mating is important for genetic studies per se, because that shed light on some seemingly inconsistent result, and Alex spoke about some of them before. So the reason why partner look-alike are elusive, but here I've just listed three well-accepted reasons. So the first one is social homogamy, which simply refers to the fact that you're more likely to find your partner within the social groups that you attend. So if you are into music, you're more likely to find your partner at a concert, for example. If you are into sport, you may find your partner at the gym. If you are into mathematics, I wasn't sure if that joke would work here, but apparently it did. So another reason is a sort of a direct choice driven by established criteria, as I like to term it. So I know people who have a precise list of criteria they're looking for, including looking for some partner that exactly look like them. And a third one, which is less related to mate choice, is convergence in time. So you can think about people being together for a long time, and after a few years or few weeks, they start to converge to some similar trait, for example, body mass index. So what we'll talk about precisely today is direct choice based on established criteria, and the reason behind that choice is because it has been shown in theory to have evolutionary consequences. So those evolutionary consequences go back to foundational works from Fisher in 1918 or Wright 1921, and all this, and two others, well, actually, there are not many, I can cite them, so Krohn, Kimmerer, and Gimmelfarb. And all these theoretical works point to assortative mating creating a positive correlation between trait increasing alleles. So that will, and we're going to think about it as a signature of assortative mating, even on alleles that are on the very distant position on the genome, distant loci, sorry, the assortative mating is expected to create that correlation. So what is the consequence of that correlation? One is that it increases in the population homozygosity for trait-increasing alleles, but that has been shown to be negligible, especially for polygenic traits. But most importantly, it increases the genetic variance in the population as compared to a population where mating is at random. So what I will talk about, I'm going to talk about our study that has been published earlier this week. And this study aims at quantifying the signature of assortative mating. And as I said before, that signature is that correlation between trait-increasing alleles. So briefly, I'm going to go through the method that we developed and we implemented. I will, in the second time, talk about some of the analysis that I've run on basically applying that method to a collection of 32 traits, and finally discuss some of the challenges we had to face. Okay, starting with the method. So, third time, what is the signature of assortative mating? It is that characteristic correlation between trait-increasing alleles. But if we want to quantify the correlation between trait-increasing allele, we need to have some sort of a priori knowledge of the direction of association between one particular allele and a trait. And the way we decided to inform that so sort of direction of association was through the use of summary statistics from published GWAS. And the second idea, key idea underlying our method is that in a population undergoing random mating, we expect the chromosomes to segregate independently. So therefore, if you consider allele counts on different chromosomes, you expect those allele counts to be completely uncorrelated. While under assortative mating, as I said, we have this correlation, but thinking in terms of contrasting different chromosomes is also a nice idea because it allows us to go around linkage disequilibrium, which you can have within a chromosome. So these are the main ideas. So what we do is that we start by collecting, say, a set of summary statistics, and these are the usual type of information we have. We have the chromosome, we have the SNP ID, we have the allele that has been tested, and most importantly, we have the effect size of that allele. So then what we do is that we go into independent population, independent of the study that generated that GWAS, that summary statistics. And in that population, we create polygenic scores for individuals using SNPs on the odd chromosome and SNPs on the even chromosome. And subsequently, we try to quantify the correlation between, so each individual will therefore have two values, the polygenic score on the odd chromosome and the polygenic score on the even chromosome, and argue, and I will show you in the next slide, that correlation tells us something about assortative mating in a population. So we've done some theoretical work, and here I just put this summary equation that predict what is that correlation exactly trying to measure. So this theta parameter covers multiple things. One is that it linearly increases with the spouse's correlation. We can also show that it increases with the size of your discovery GWAS. So if you want to quantify assortative mating with this method, and you use, for example, a GWAS with say you use two GWASs, one with a small sample size and one with a large sample size, we can predict that you will get a larger theta with the one with the larger sample size. So that's also, and finally, we also show that this theta is smaller for highly polygenic traits. So we also identified two main sources of biases. One which I briefly alluded, which is that, well, sample overlap is definitely can create that correlation. So the simple fix is look for independent data set. That's a very simple fix. We could have done something a bit more elaborated to correct for that sample overlap bias, but we just wanted to start it with something very straightforward. And the second one is population structure. So population structure is a very vague concept, at least to me. But interestingly, assortative mating and population structure are essentially the same thing, right? You can think about population structure as a sort of geographical assortative mating. You tend to find your partner, well, within a certain kilometer radius around where you live. So it's therefore easily conceptualized that it's trying to estimate assortative mating using the polygenic score strategy can be easily confounded by population stratification. So what we did as a way to correct for that bias was simply to adjust for principal components, as people do generally, and we found here that it works pretty well. Okay, so now I'm just going to present some of the results we basically applying that method to a collection of traits. So I've analyzed 32 traits from basically everything I could find in the literature. So for this independent, before that, so for all the traits, I basically selected variant that showed some evidence of association with the trait. So that's why I selected SNPs with a p-value below 10 to the minus 5, 10 to the minus 3. We can discuss that arbitrary choice if you want. And also, I've selected independent variants, and that's something that we found to be important as well. So we considered 3 independent data sets: the UK Biobank, JERA, which is an American cohort, and the Health Retirement Study, which has been presented today. And also, I'm going to present some of the results that we did, what I call the verification analysis using 18,000 spouse pairs in the UK Biobank. So at this point, before showing the next slide, I just want to highlight that what we're trying to do here is to estimate the signature of associative mating without using spouses. And I think that's the main feature of our method, is that you can learn what happened in the population in terms of mating without actually observing spouses. So, well, it may not be the say a reasonable assumption, but if you can just, as a thought experiment, think about in 1,000 years when if humans will be on a different planet or completely wiped out, that some other species or some future humans will basically collect DNA and try to learn something about what was the mating like in the 20th or 21st century. And using a method like that, and hopefully they will cite my work, they will be able to tell that there was actually associative mating in the population, and that associative mating was driven by similarity on height, on education, and, well, I guess these are the two things that we found, actually. So among the 32 traits, we found significant evidence of associative mating for height and education. So it was sort of reassuring to find those usual suspects. And I just want to say that we also consider a null trait, which is bone mineral density, for which we knew that, well, we clearly don't see a correlation between bone mineral density of spouses. And and it was also nice to find that with this method, when there is no associative mating, we're also specific on that point. So we also wanted to verify that those findings, and so in our theoretical derivation, we predicted that the theta parameter that we estimate is actually half of what you will get, you would have estimated if you basically correlated polygenic scores of spouses. So what you're looking at here on the x-axis is that theta parameter that I presented before, that we estimated just in a collection of unrelated individuals. And on the y-axis, you have the actual correlation between spouses that we calculated using 18,000 spouse pairs in the UK Biobank. And so the predicted line here has a slope of 2, and what we observe is that it's pretty consistent, although it's being trying to fit a line with two major points. But interestingly, I also argue that we might have missed some evidence, some traits, simply because the current size of GWASs for those traits are basically smaller than for height and education. So for example, there is this particular dot. We can actually see the mass. So this one is intelligence. So it's a smaller GWAS, but we also have evidence of correlation for IQ between spouses. So we could expect that with a larger GWAS, that point will probably move a bit up. So now I'm going to talk about some of the challenges, and I briefly mentioned that population structure is one of them. So in this particular study, we really wanted to be conservative in a way that we didn't want to claim associative mating when there was not associative mating. And how we addressed that was by being very stringent with our PC correction. And I'm pretty sure that by doing that, we potentially also have removed some interesting signal, which we may subsequently address with a larger GWAS, hopefully. And one more time, one of the opportunities of this study was to basically learn something about associative mating without the need of spouse pairs. And I think that opens very interesting avenues of investigation. And in particular, mate choice is a complex object. It involves height, it involves education, but it probably involves so many things. And for those things, it's likely that, well, over the years, we will have, for many traits, very large geoses. And with a method like that, we can, without any a priori, try to interrogate, is there any similarity between spouses for things that we haven't measured in most of the studies where we have actually observed the spouses. And one thing, it's a bit of, actually, I'm not sure where I want to go with that, but I feel like we could also try to dive a bit more in the molecular space. And I was thinking about maybe we could address, is there any similarities for gene expression or for other molecular traits that we can measure? And I hope that by looking at those very basic traits, that somehow we will have, we could gain insights into what are the sort of higher level traits on which people sort in the population. So those are my final words. So I'd like to thank my co-authors who are listed here, in particular Peter Vischer, who's been my mentor for 2.5 years. So it's been great working with you, Peter. And also the funding bodies. So thanks for your attention.
Questions for both papers, which I'm really looking forward to reading. For Loic, so what you're really detecting in the genome is a sort of mating of the parents. So have you looked at all, maybe this explains the slight differences, whether or not there's changing in a sort of mating by marriage year or something, so that those folks that you can actually observe in UK Biobank who are older more match, maybe you're underpowered for this, more match the estimates from the genome of essentially the parents. I mean, that it could be a generational difference that you're picking up, and there could be also ascertainment bias if the alleles and the strength of the alleles, the strength of the betas differed from early in the 20th century, say for women when there was limited educational opportunities, than later in the 20th century. So how you think about sort of change over time. Should I throw in my second question? And for Alex, the question is, did you look at all for any evidence of non-violation of Mendelian inheritance? I mean, Molly Jaworski has that paper from a few years ago that looked for sites in the genome that showed bias. And I don't know if you've looked more generally, because if the bias is induced from environmental differences like stress, let's say, in utero, then they would, I would reason, represent a violation of the TDT.
Yeah, thanks, Dalton. Yeah, absolutely right. What we measure here is the assortative mating in the parental generation. So does it change over time? So I've been actually discussing with Patrick after the paper was out. I've also looked at, basically, you have this information, the score on odd and even, and if there is any change over time, I was thought about, well, that should correlate with the year of birth of those people. And what we see is that there is no correlation with height, but there is a positive correlation with education. So it seems like assortment on education has increased between the 30s and the 70s in the UK population. Yeah, so there is something funny actually. Well, thanks Peter for pointing that. It's I've run this analysis when you basically you accumulate, you estimate the theta by, for example, starting with people born before 1940, people born blah blah, etc., until you include the entire cohort, and you have a very interesting shape where the estimates start to climb, to go up, and drops around 1945, surprise, and then goes up again. So my take on that was there's probably something about the World War. Maybe people are less picky in wartime, but I don't know. Yeah, that's it.
So yeah, I did actually look in trios in the decode data for violations of Mendelian inheritance, and if you look genome-wide, then it's quite amazing how close it matches the predictions. I mean, there are going to be violations of Mendelian inheritance when you have like recessive lethals. You're not going to observe them in your data. But I think genome-wide, the violations from Mendelian inheritance are going to be really very minimal. I think it's about as robust an assumption as we ever get in biology. So I doubt this is going to affect the sort of complex polygenic traits that we're looking at here much. Maybe if we're looking at rare disease traits, it could be more of an issue.
I actually have a question for both Melinda Mills and Loic Yengo. So there are other evolutionary forces that could induce linkage disequilibrium or gametic phase disequilibrium between distant sites in the genome. One of these is actually natural selection. There's a so-called Bulmer effect, which says that under certain forms of epistasis, different sites affecting the same trait will actually be induced to show negative linkage disequilibrium. And this actually, the existence of such an effect is actually the basis for the Fisher-Muller hypothesis, class of theories for the advantage of sexual over clonal reproduction. And so my question is, so earlier it was mentioned that there's a new GWAS of years of a total number of children that's coming out. That's pretty good for actually total fitness now. So my question is, what is the sample size for that going to be? And then, according to the equation for theta, do you have a feeling about whether that will be adequate to detect a negative gametic phase disequilibrium if such actually exists?
I'll let you answer the question directed at you, but the sample size, it depends because we have age restrictions. So age at first birth, number of children ever born, people have to reach the end of their reproductive period. So 45 and up for women and 55 and up for men. So it's around 800,000. And it depends on the different phenotypes. So maybe we'll get on one of your dots eventually there. And then before I pass it on to you, I'm just going to ask a question to you as well too. So there's this whole area called sociology that's studied assortative mating for many years, and we find exactly the same thing. That there's assortative mating on education and height. So maybe you could tell us a little bit more about what the genetic side of it adds to that. But you have to answer his question first.
Actually, I'll start with yours, which seemed to me a bit easier. I think that the genetic basically trying to tease apart the three things that I presented. So when you observe a correlation between spouses, well, that could as well being driven by mate choice, and in this case, we should pick that up with genetics. While what just looking at the correlation won't tell you if that happened. Well, I think for height is probably easy to think about. But for example, for psychiatric traits, there is this evidence of spouses being correlated. They have a correlated liability for traits like schizophrenia. And is there, well, unfortunately, we didn't have enough power to detect that, but that was one of the driving questions for this study. So for James, is that James, right? Yeah. So I think in theory, that theta parameter can absolutely detect positive or negative correlations. I think when it comes to natural selection as compared to assortative mating, I feel like the timescales are a bit different. So the assortative mating will basically, you will build that correlation fairly quickly. Within one generation, you can have a massive effect. Whereas I think the strength of selection will need to be very strong to be detected just within one generation. So I feel like this effect of the theta that we detect here is mostly dominated by something that happened in the more recent past. I think Peter wants probably add something on that.
I have a question for Alex, who's linking the two talks. So under assortative mating, there will be a correlation in the effect of the transmitted and untransmitted allele, as well as a correlation between the effects of the transmitted or untransmitted alleles between the two parents. When you illustrated your different estimators of heritability on your nice graph with IQ, you mentioned that it's likely in humans that the environmental shared environmental effects go up with increased relatedness between people. But if you actually looked at the correlations that you had on that for IQ, and you would do a most naive analysis, which is multiplying the phenotypic correlation or dividing it by the relatedness, then you would find that your estimate of heritability, so for half-siblings, that's 4 times the observed correlation, for cousins 8 times, you would come to the conclusion that the distant relatives are disproportionately similar to each other. And that is what you would predict under assortative mating. So I'm just wondering how assortative mating confounds your inference from your method for those kind of traits.
Well, I'm not sure it confounds the inference for my method, but I think the question was more about how it might affect the other kinds of methods, because I'm using the deviation in the relatedness, which is going to be quite robust to that kind of pattern where assortative mating makes the correlation between relatives fall off more slowly with relatedness, but I don't think that would apply to the deviations around the expected relatedness. I think that would be captured in the expected relatedness from the parental side. But yeah, I think the IQ thing is interesting because, I mean, if there's assortative mating on education, then there will be assortative mating on IQ. And I think there's an interesting question of whether some of these results we have for educational attainment with the indirect effects being important, do they also translate over to IQ? I don't think we know the answer to that yet.
So I wanted to propose an answer to this question of what does this add to everything. So one of the things is if you see changes in the similarity of the genes, so the genotypic assortative mating, if you will, there are kind of 3 possibilities. One is just the population stratification is different and who people are hanging out with is different. Another is that you have a G by E change where the expression of the genes into the phenotype is different, and therefore you have more or less assortative mating if the assortative mating is based on the phenotype. And then the third thing is the phenotypic assortative mating changing. So if you combine kind of more regular survey data, or the kind of data that the sociologists look at to see what's happening with the phenotypic assortative mating, with this gene data, you can now do kind of a decomposition. There's a change in the correlations of the genes. How much of it was because people with that phenotype actually got married or whatever together. The other thing that the assortative mating, that the gene data on the assortative mating does, is in principle it can reach back in time to times when we didn't have the data we didn't have the sociological type data. So you basically learn about assortative mating at the time of somebody's birth. So if you have data on anybody for their genes and you look at the variance, then and we don't always have the sociological data we would want for the assortative mating at the time of people's birth who are in our genetic sample.
So something, I think this is for Alex, but something that struck me as being interesting about the pairing of these talks is so we would often think of the spouse as being the most consequential person in somebody's life who is by norm and usually by law, right, not allowed to be a blood relative of theirs, right? And once you open up the idea of genetic nurture to allow siblings and such, then you start to think, well, of course, spouses, we know spouses are more like each other, right? We know friends are more like each other. We know neighbors are more like each other. We know almost everybody that sociologists would think of as having an influence on somebody's life is more likely to be genetically related versus a random person, right? And so then one would think, well, maybe like is virtually every social environmental effect that involves some kind of interpersonal effect, really in some ways we would think of as being an indirect genetic effect. And that train for me would halt abruptly, perhaps with the idea that, well, but the extent to which two friends are genetically closer to one another, so it's still nothing like the way third cousins are genetically closer to one another. But the other side of that would be that, well, if we're talking about the polygenic score on any trait, if we're talking about polygenic information, that would no longer, I would think, be the case. That correlations would be appreciable and closer to the phenotypic correlation. And I guess for how you're thinking about genetic nurture, I'm wondering which of those two things I should think about. Should I think, well, this is kind of a nuisance issue because the actual friend genetic relatedness is really not that big compared to family relatedness, versus the idea that, but if we're thinking about genetic nurture in more polygenic terms, this could actually be an appreciable force.
Well, I think the key thing is the coefficient of relatedness of the person who's influencing you through the environment. So I think that there will be some contribution from friends, but in general it's going to be quite negligible compared to the contribution of the parental indirect effects or sibling indirect effects to any kind of association statistic that you're seeing with the genotype of the proband. It's going to be interesting to investigate that more. I've seen some papers showing greater relatedness for friends, but I can't see it contributing majorly to a lot of the issues that we've been talking about.
Can I just quickly add to that?
Yeah.
So I think your method is robust. To the extent that we consider people selecting into their environments and then getting that environmental exposure as part of G, because they're seeking out those friends, then that's fine. All my friends are geniuses around here. But to the extent that if it were somehow dictatorially imposed by your measuring your genotype and then there might be some scenarios where it's strange. There's some weird effects, but with the sibling difference or the transmission disequilibrium writ large across all the types, I don't see that as a problem, because the key thing you're trying to estimate is randomly assigned. So even if it ends up that you select into an environment with different genotypes, so what? That's part of the effect. Unless I'm misspeaking for you, I don't know.
Well, sorting with people that are similar to you is in some sense an effect of your genotype, right? Yeah.
I think in this discussion, you have to be very careful what you mean exactly by genetic relatedness. So if I have a very large population, very large effective size, so everybody's unrelated kind of to each other, and there's assortative mating on a trait like height, I expect let's call it the genetic values, or what you're trying to get at with the polygenic risk score, they'll be correlated by 0.2 or so. But that's nothing to do with their IBD relatedness. It's just because the genetic values are correlated. It's not the same relatedness as cousins or half-sibs.
Yes.
Thank you. Those were both very interesting papers. I had a question for Alex Young. Have you been able to at all look at whether these measures of genetic nurturance kind of oscillate throughout the life course? So I would expect the estimate to be higher maybe when a person is, in primary school in terms of measures of school achievement. But then when they go off to college, does the proportion that's due to genetic nurturance go down relative to the person's own kind of, heritability of certain traits?
The short answer to that is no, we haven't been able to look at that, but I'm sure there are differences. If you look at twin studies, you do see that the shared environmental influence varies depending on the age at which you measure like educational phenotypes. So I imagine something like that would be mirrored in the genetic nurture results, but we've been restricted mainly to this measure of educational attainment, that's years of education. So that's sort of seeing when someone's completed their education.
So do you think that might have biased it downwards
Well, I'm not sure, because that years of education phenotype might be particularly prone to have these strong familial influences, because how many years of education you complete is partially a function of support that you get from your family to complete that, whereas school grades might be more directly related to something like IQ, where maybe this influence is smaller. So I think it depends exactly on what educational measure you're looking at.
So I have a question, or rather comment, more targeted towards Alex's paper. So just what's very interesting about the genetic nurture research is that we find, heritability, as traditionally estimated, should be a bit lower. But the flip side of that is that one's gene impact more than just one's traits. So we can think of this concept of, I would say, like genetic externalities. So the parents' gene impact their kids, but also there's lots of peer effects, which are multiplied over many more peers than just the kids. And so I guess it's just a question, have you thought of this at all, or any, yeah.
Yeah, well, I mean, I think to some degree it's interesting to think about the overall influence of genetics in generating sort of inequalities between families. And these kinds of indirect effects from relatives can increase this sort of the genetic contribution to like between family inequalities, even if in some sense you might think some of these results are saying that genetics is less important than you might naively assume from just looking at correlations of polygenic scores. I actually don't think that's quite the right way to interpret it. It's more that the causal path from genetics to generating differences between people is more complicated. And one of the interesting things about these nurture effects that go from parents to offspring is it means that the trait distribution is not stable. And in some sense, these effects accumulate over generations before they reach some sort of equilibrium. So I think I actually suspect that part of the reason that you see population stratification and gene environment correlation is kind of the result of these indirect effects iterated over many generations.
And also peer effects in the neighborhood. It's more than just within families.
Yeah, because the relatedness is sort of clustered with geography. Yeah.
It's pretty quick. It'll be for Alex. I was just curious. So in the initial, or it could be for Augie too, but in the initial genetic nurture paper in Science, with EA2 is what you guys used. You couldn't reject. So this null that the male and maternal and paternal effects were identical, but there's a pretty wide confidence bound there. Yeah. So I was just curious, EA3's out. You should be more powered. Have you guys taken a second look at that?
I mean, that would require access to the same data that we had, and that's not necessarily forthcoming. I mean, I think that the sort of null hypothesis test is uninteresting to me, because in nature, nothing is ever symmetrical without a very solid reason to be symmetrical, and there's no reason to expect symmetry. So it would be interesting to measure this more precisely, and I think that will happen sooner or later.
I just want to add one thing. Yes, indeed, partly because I think it will be politically correct and so on. So of course in the paper, of course it happened to be really true that the mothers do contribute more to the health of the kid, right? But actually there's one result that indeed we didn't publish. There is one trait where the father is stronger than the mother. And that happened to be chess playing. So basically, again, it's again just using the educational attainment polygenic score, because it happened about paper. Even because in Iceland, chess playing is very popular. I mean, the Fischer Spassky match was held there, but still, even then, of course, there are women who play chess, but still, it's mainly men, and so it's mainly passed on from men to sons, and so you can really see it there. Yeah.
Cool. Thank you.